Hello dear students, myself Bhavani Pentela, working as an assistant professor in Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. In today's lecture, we are going to discuss about G protein coupled receptor. So without any further delay, let me dive into the session. So in the previous lecture, we had discussed about the transduction mechanism and how the transduction mechanism will be seen, and after of that we had explained about the ion channel receptor. So what are the ion channel receptors and how the channel is going to open and how the channel is going to close, and there are some drugs which can open the channel, there are some drugs which can close the channel. For example, acetylcholine is a drug which can open the sodium channel, D-tuberculonin is a drug which can close the sodium channel. If I am talking about nicotinidyl is a drug which can open the potassium channel. There are some drugs like verapamil, diltiazem, these are the drugs which can close the calcium channel. There are some drugs which can open the calcium channel. So depending upon the drug, what the channel is going to open and close will depend upon the what the drug you had administered and what drug is going and bind to the particular channel. And in today's session, we are going to discuss about the Z protein coupled receptor. So what is any receptor, which I had discussed in the previous lecture as well? Receptor is a protein, it is a macromolecule which is present on the cell surface and which is present inside the cell as well. And once the chemical molecule bind to its receptor, it will shows its action by the binding of the drug, and itself do not having any action. If I am talking about the receptor itself do not having any action. Once the drug bind to its receptor either on the surface or inside the receptor, there will be some changes happen inside the receptor and which will causes the drug receptor complex and which will causes the physiological actions or pharmacological actions. The drug may be the neurotransmitter, it may be the receptor, it may be the hormone, or it may be the any substances. When it bind to its receptor, it will show its action. So now we will discuss about the GPCR receptor in shortly, which you can call as an Z protein coupled receptor. Z protein coupled receptor you can call as an serpentine receptor, you can call as an GPCR receptor, you can call as an 7 transmembrane metabotropic receptor, 7 transmembrane helical, because it having the 7 transmembrane helical in structure. So it is called as a 7 transmembrane receptor. You can call as an a metabotropic receptor. So ionotropic is different with that of a metabotropic. Sometimes the GPCR is can call as a metabotropic receptor. So which type of receptor it is? The Z protein coupled receptor is a cell surface receptor. Why you can call it is a cell surface receptor? Because the receptor which are present on the surface of a cell. Here the drug can bind to the receptors which are present on the cell surface area. Okay. So now we will see the importance of an Z protein coupled receptors. Why the Z protein coupled receptors are important? If I am talking about this Z protein coupled receptor, this is it plays very important role in maintaining the homeostasis. It plays an important role in growth and metabolism. It will activate the T cells. It will plays an important role in behavioral changes. It will plays an important role in mediate the senses like smell, vision, taste and pain. Okay, it also plays an important role in inflammation. So this is the main importance of an GPCR. It plays an important role in inflammation, it will activate the T cells, it will mediate the senses, it plays an important role in behavioral changes, it will plays an important role in metabolism, growth, and it plays an important role to maintain the body homeostasis. Okay, so these are some examples which falls under GPCR. Most of the receptors, most of the receptors present in the body will act through the GPCR. It is a large family receptor compared to that of a rest of receptor. So if I am talking about the this GPCR receptor, there are some few examples which falls under this receptor. If I am talking about the muscarinic receptor for acetylcholine is an GPCR receptor. Adrenergic receptors, both alpha and beta are the GPCR receptors. And if I am talking about the opioid receptors are also in a GPCR receptor, adenosine receptors, histamine receptors, serotonin receptors, except 5-HT3. Except 5-HT3 is all rest of all receptors are GPCR receptors. And GABA receptors, forget about GABA A and GABA C. GABA B is a metabotropic receptor, it is a GPCR receptor. Dopamine receptors, glucagon receptors, metabotropic glutamate receptor, if I am talking about the glutamate having the 3 types of receptors. One is the metabotropic glutamate receptor and inotropic glutamate receptor, NMDA, AMPA. It will falls under inotropic receptors. There are some metabotropic glutamate receptors as well. And if I am talking olfactory receptors, this is also GPCR receptors, prostaglandin receptors, right, angiotensin receptors, bradykinin receptors, all these receptors will falls under GPCR receptors. Okay. So, if I am talking about here the muscarinic, so here I have mentioned the muscarinic because acetylcholine having the 2 types of receptors, one is muscarinic and second one is nicotinic, and muscarinic will falls under GPCR receptor, but not the nicotinic. And if I am talking about adeno receptor, both alpha and both beta are the GPCR receptor. Okay, opioid receptors, all opioid receptors are the GPCR receptors, right. And next one we will discuss in detail about the Z protein coupled receptor. So, what is a Z protein coupled receptor? Z protein coupled receptor is a 7 transmembrane spanning hydrophobic amino acid segment. So, if I am talking about here, it is it having the 7 transmembrane. So, see here 1, 2, 3, 4, 5, 6, 7. It is having the 7 transmembrane receptor. It is a 7 transmembrane helical. It having the 7 transmembrane helical in structure. So, if I am talking about here, it having 6 loops. So, see here, this is the 1 loop, 2nd loop, 3rd loop, 4th loop, 5th and 6th. In GPCR, we having the 6 loops which consists 3 loops in extracellular and 3 loops in the intracellular. So, if I am talking about this is a GPCR receptor, this is an extracellular and this is an intracellular. The N terminal which is present in the extracellular and C terminal which is present in the intracellular. So, we having the 6 loops in GPCR, the 3 loops which are present, this is 1 loop, 2nd loop and 3rd loop. The 3 loops are present in the extracellular and the 3 loops are present in the intracellular. Okay. So, the signal molecule will be present some other some surface of this alpha helical structure, and there is an intracellular domain having the Z protein receptor. So, this is the reason why the receptor called as an GPCR receptor. So, this G protein coupled receptor, if I am talking about the drugs, 30 to 40% of a marketing drugs will act through the GPCR receptor. It is the main target of the particular drugs by acting on GPCR receptor. The GPCR receptor are the cell surface receptor. It is in a large family of any membrane, cell membrane receptor. And if I am talking about Z protein coupled receptor, it has been falsified into alpha receptor, beta receptor and gamma receptor. And this alpha receptor is further classified into GS, GI, GO and GQ. What is GS? What is GS? G protein stimulatory. So, here when the drug bind to its receptor, depending upon the drug, the action, the G protein coupled receptor will shows the stimulatory, GI inhibitory, G protein inhibitory. And what is GO? GO represent here the calcium channel decreases, GQ phosphokinase, phospholipase, the phospholipase will get increased. Increase in phospholipase we can call as an GP. Okay, GI stimulatory, GI inhibitory, GS stimulatory, GI inhibitory, GO decrease in calcium channel, GQ phospholipase. So, if you see here the GPCR signaling having the 2 main pathways, one is adenyl cyclase pathway and second one is phospholipase pathway. So, if you see here GPCR, this one is by the adenyl cyclase pathway, this one is an a phospholipase pathway. So, first we will discuss about the adenyl cyclase pathway. If I am talking about here, this is an GPCR receptor, right? This is an GPCR receptor, which will shows its action by the stimulatory or inhibitory. If the drug bind to this G protein coupled receptor, if the adenyl cyclase activity get increased, then what is going to happen? Then the protein kinase will be get stimulated. Once the adenyl cyclase activity increased, how the adenyl cyclase activity increases? Once the adenyl cyclase activity increases, it will convert the ATP. What is the function of adenyl cyclase? Adenyl cyclase function was it converts the ATP to the cyclic AMP levels, and this cyclic AMP level in turn it will activate the phosphokinase A. And what about the phospholipase pathway? The phospholipase pathway is a GQ pathway. Here it will activate the phospholipase, and where you can in turn activate the phosphokinase. Here we can activate the phosphokinase, which I will explain in detail in the forthcoming slides. Okay, now we will see what happens when the drug binds to the Z protein coupled receptor. So here you can see the two pictures which represent A and B. The first picture where it shows there is no drug binding, and in the picture B you can see the drug binding. So we will discuss here when the drug binds to the receptor, and if there is no drug to the receptor, what is happening inside the GPCR. So if you see the first receptor here, there is no ligand binding to this receptor. If you see here there is no ligand to its receptor. So which I had said you in the previous slide, this is a 7 transmembrane helical structure, 1, 2, 3, 4, 5, 6 and 7. This is having the 7 transmembrane helical structure which consists of 6 loops, 3 intracellular and 3 extracellular. It consists of a ligand binding domain, and inside it consists of a Z protein receptor. Okay, if there is no receptor occupied by the drug, if there is no drug bind to its receptor, see here, see in this picture there is no drug. If there is no drug bind to its receptor, then the Z protein will be coupled with the GDP. Then the G protein alpha, beta, gamma will be attached with the GDP, right. If there is no drug bind to the receptor, Z protein coupled receptor, the alpha, beta and gamma subunit will be bind with the GDP. And see the second picture here, the drug has been occupied. Once the drug has been occupied to its receptor, once the drug can come and bind to its receptor, then what are the internal changes will be happen? Once the drug bind to its receptor, here alpha subunit will be separated from the beta and gamma, and GDP will be replaced by the GTP. See here, the GDP will be replaced by the GTP in the presence of guanine nucleotide exchange factor. So which factor is replacing the GDP to GTP? Guanine nucleotide exchange factor. So don't get confusion here. When there is no drug bind to the receptor, the G protein alpha, beta and gamma will be attached with the GDP. Inactive state of an receptor. Once the drug bind to its receptor, what is going to happen here? Once the drug and gone bind to its receptor, here the conformational changes will be seen inside the receptor. The alpha subunit will be get separated to its beta and gamma, and GDP will be replaced by the GTP by the guanine cyclase nucleotide exchange factor. Now this GDP has been replaced. So when the GDP is attached, it is an inactive state, there is no drug. So the G protein is attached with the GDP. Here the drug bind to the receptor, and what has happened? Once the drug bind to its receptor, once the drug has been bind to its receptor, here the GDP has been replaced with the GTP. So once the GDP has been replaced with GTP, here you can see the two pathways of an Z protein coupled receptor. The two main pathways are cyclic AMP pathway, which you can call as an adenyl cyclase pathway, and second pathway was IP3 DAC pathway, which you can call as an phospholipase pathway. So these are the two pathways, these are the two important pathways which you can see in the GPCR receptor. So now we will discuss about one by one what will be happen in the adenyl cyclase pathway and what will be happen in the phospholipase pathway. So first we will discuss about the adenyl cyclase pathway. So this is an adenyl cyclase pathway. So what I had said you in the previous slide, I had said you when there is no drug bind to the receptor, the Z protein will be attached to the GDP, right? And when the drug bind to the receptor, the GDP will be replaced with the GTP, and alpha subunit will be bind with the GTP, beta and gamma subunit will be get separated, and this will activate some other proteins inside the cell, and this alpha will be get separated, beta and gamma will be get separated, right, which I had shown you here, right. See here, this is the receptor. When the cell has been activated with the drug, the alpha has been separated with the beta and gamma, and what this beta and gamma will do? The beta and gamma will activate some other proteins inside the cell. So let us see here adenyl cyclase pathway. What happens in the adenyl cyclase pathway? Adenyl cyclase pathway, once the drug didn't bind to its receptor normally, then the cyclic AMP levels will be not activated. If there is no activation of cyclic AMP levels, the regulatory units and catalytic units will be in the bound form, and there will be no cellular changes will be seen inside the receptor. Once the drug come and bind to its receptor, the Z protein alpha receptor will be get separated to its beta and gamma, and GDP will be replaced with the GTP, right. So if you see here, this is an GPCR receptor. which G protein, it consists of any 3 units alpha, beta and gamma, right. When the drug bind to its receptor, the alpha subunit will be get separated, GDP will be replaced with the GTP. Okay, GTP. Now, the alpha receptor will be get separated and which will be replaced by the GTP, and this GTP of the GABA GTP of an alpha receptor will activate the adenyl cyclase enzyme. Adenyl cyclase is an enzyme which is present somewhere on that membrane of an receptor, and once the alpha receptor has been activated, when GDP has been replaced with GTP, then this activated G protein will activate the adenyl cyclase enzyme. And what is the main function of adenyl cyclase enzyme? The adenyl cyclase enzyme will convert the ATP to the cyclic AMP levels, right. Now, the adenyl cyclase enzyme has been activated, and what is the function of this enzyme? It will convert the ATP, it will convert the ATP into the cyclic AMP. So, these are the cyclic AMP. So, see here, these are the cyclic AMP levels, and these cyclic AMP levels will in turn activate the phosphokinase A. So, phosphokinase A, what is the role of a phosphokinase A? What is the function of phosphokinase A? It will phosphorylate. So, you can see the phosphorylation, it will phosphorylate the transcription factor. Okay. So, normally this phosphokinase A in the cell, it will be in an inactive state. So, if there is no activation of adenyl cyclase, there will be no activation of an pKa. Normally pKa will be in inactive state, it consists of 4 units, like 2 regulatory units and 2 catalytic units. So, see here, so this is an phosphokinase A, this is an phosphokinase A which is present inside the body in inactive form, which consists of 2 regulatory subunits and 2 catalytic subunits. You can see here, C represents the catalytic, R represents the regulatory. So, the phosphokinase A which is present in inactive state, which consists of regulatory units of 2 and 2 catalytic subunits. When these catalytic subunits bind with the regulatory subunits, then it cannot show any catalytic action. And what will be happen once the adenyl cyclase activity has been activated? This adenyl cyclase enzyme has been activated. The ATP will be converted into cyclic AMP, and this cyclic AMP, see here, this one is the cyclic AMP. This cyclic AMP will go and binds to the catalytic. It binds to the regulatory unit of an phosphokinase A, and when it binds to the regulatory units of an phosphokinase A, the regulatory unit will be get separated from the catalytic unit. So, see here, so the cyclic AMP levels has been separated the regulatory units. When the regulatory units has been attached with the catalytic unit, it cannot show any action. So, here the regulatory units has been separated by the adenyl cyclase. This adenyl cyclase is a cyclic AMP. The cyclic AMP levels will separate the regulatory units, and these regulatory units now has been separated and catalytic units has been separated, and these catalytic units will activate the further proteins. It will phosphorylate the further proteins. It will phosphorylate the further proteins present, and this catalytic subunit will enter inside the nucleus, and there is an important protein which you can call as an CRB, and this catalytic unit will phosphorylate the transcription factors. Once it has phosphorylated the transcription factor will get activated. So, this is an adenyl cyclase pathway. So, have you understood about the adenyl cyclase pathway? It is a very simple pathway. Once the receptor has been occupied with the drug, the G protein will be get separated. G protein in sense alpha subunit will be separated with the beta and gamma, and GDP will be replaced with the GTP in the presence of guanyl cyclase, guanine exchange nucleotide factor, right. And when it has been exchanged by the GTP to GTP, and this GTP will in turn activate the adenyl cyclase. The adenyl cyclase activation will causes the conversion of ATP to the cyclic AMP. Once the cyclic AMP has been converted, this cyclic AMP in turn will activate the phosphokinase A. In turn it will activate the phosphokinase A. Normally the phosphokinase A in the body which is present in a inactive state, it consists of 2 regulatory units and 2 catalytic units. When the catalytic units bind with the regulatory units, it cannot show any cellular effects. So, this cyclic AMP levels, the cyclic AMP levels, here the cyclic AMP will come and bind. So, what the cyclic AMP will do here? The cyclic AMP will activate the phosphokinase by removing the regulatory subunit from the catalytic catalytic subunit. It will separate the regulatory subunits. When the regulatory subunits has been separated from the catalytic subunit, now these catalytic subunits will phosphorylate the other proteins and it will enter inside the nucleus. There is an important protein which you can call as an CREB. So, this is a protein CREB and which will phosphorylate the transcription factor and which will causes the transcription factor activated. And next one is phospholipase pathway. So, what is a phospholipase pathway? Phospholipase pathway is a second pathway of an Z protein coupled receptor. So, if you see here students, so the same way the GTP is converted, GTP is being replaced by the GTP till the same, and now we will see how the phosphokinase pathway will be get activated. So, say here this is an so see here I had forgotten to explain you. See here, this is an the primary signal was the primary signal produced to the receptor was an ligand, and what is the secondary signal? Cyclic AMP. The secondary messenger, what is the secondary messenger here? Cyclic AMP. Cyclic AMP is a secondary messenger, it is a secondary signal. So, if you see here students, the signal molecule you can call as a first messenger, because it is the first signal molecule which is going to activate the receptor, right. When the signal molecule come and bind to the GPCR, so this is a GPCR receptor. When the signal molecule come and bind to this GPCR, then what is going to happen? The GDP will be replaced by GTP. Now, the G protein has been activated, and this activated protein will activate the phospholipase C enzyme, and this phospholipase C enzyme, what it will do? It will hydrolyze the IP3 and DAG. It will hydrolyze the IP3 and DAG, and this IP3 will go and bind to the sarcoplasmic reticulum of a cells of a channel, and when it bind to its channel, the channel will be get opened and the calcium will be entered from the sarcoplasmic reticulum to the cell. Now, inside actually if you see, this is a sarcoplasmic reticulum. Okay, inside the sarcoplasmic reticulum you can see the calcium. Once this IP3 come and bind to its channel, the IP3 will open, it will initiate to open the channel. Which type of channel it is going to open here? It will open the calcium channel. Once the calcium channel has been opened, the calcium will move out from the sarcoplasmic reticulum, and this calcium will combine with the DAG to activate the phosphokinase C, and in turn it will activate the cellular changes. So, in the next diagram which will shows in detail about the phosphokinase pathway. So, see here students, so this is a signal molecule, this is a primary messenger, right, this is a primary messenger, and when the signal molecule bind to the GPCR, it is a 7 alpha helical in structure. When this signal molecule bind to the GPCR receptor, what is going to happen? The receptor will get activated, the G alpha protein will be get activated and it will be replaced by the GTP. Once the GTP will get activated, in turn it will activate the phospholipase C. So, what is in turn it will activate the phospholipase C. The phospholipase C will it will catalyze the reaction, it will catalyze or it will hydrolyzes the PIP2 membrane to produce the IP3 and DAG. What is PIP2? So, if I am talking about PIP2, so it will it catalyze PIP2. What is PIP2? PIP2 is a phosphatidyl inositol 4,5 biphosphate. What is PIP2? Phosphatidyl inositol 4,5 biphosphate, you can call as a PIP2. Once the PIP2 will be hydrolyzed to produce the IP3 and DAG. IP3, what is the full name of IP3? You can call as an 1,4,5 triphosphate. Okay, what is an IP3? Inositol 1,4,5 triphosphate. What is the I represent inositol. Triphosphate. What is the triphosphate? 1,4,5 is an triphosphate. Now, what is an DAG? What is the full name of an DAG? DAG represents the diacyl glycerol. So, DAG is an diacyl glycerol. What is an PIP2? PIP2 is a phosphatidyl inositol 4,5 biphosphate. Okay, 4,5 biphosphate. And what about IP3? Inositol 1,4,5 triphosphate. And what is then a DAG? Diacyl glycerol. So, what is happened here beta? So, look at here, when the drug molecule come and bind to the Z protein coupled receptor, the Z protein will be get activated and in turn will activate the phospholipase pathway. This phospholipase pathway, once it get activated, it will in turn activate the PIP2. This PIP2 will catalyzes or it will hydrolyzes the IP3 and DAG. Once the IP3 and DAG has been produced from PIP2, so this is an IP3. Okay, this is an DAG. Okay, once the IP3 and DAG has been produced, what is going to happen here? This IP3 will go and bind to the channel which is present on the sarcoplasmic reticulum. Once the IP3 has been hydrolyzed from the PIP2 layer, then what is going to happen? This IP3 will go and bind to the calcium channel which is present on the endoplasmic reticulum, and now this calcium channel will be get open. Once this IP3 will go and bind here in this channel, then the calcium channel will be get open and the calcium will be enter from the sarcoplasmic cycle. The calcium will enter out from the sarcoplasmic reticulum, and this calcium will combine with the DAG to activate the phosphokinase C. And once the phosphokinase C has been activated, there will be seen some cellular effects produced by the phosphokinase C. So, this is about the adenyl cyclase pathway and phospholipase pathway of GPCR. Thank you.